This is the next video on this cell. Okay, now mitochondria and plastids are in this video. Mitochondria was to few part of mitochondria was already done in the last video till its till its number. Now, structure of mitochondria. It is a double membrane organelle, and the outer membrane has a lipid protein ratio of 40:60. Outer membrane has porins also for transport across membrane. Outer chamber also called perimitochondrial space. Chamber is called perimitochondrial space. It is poor in enzymes. Inner membrane is physiologically active and therefore it has several named folds called cristae. Cristae has a lipid protein ratio of 20:80. It is cardiolipid, that is modified lipid for attaching special electron carriers like cytochrome and ubiquinone. And then the cristae also has F0, F1 particles or oxysomes. So these are also known as cristae as following enzymes: ATP synthetase and succinate dehydrogenate. Now the fifth layer, that is inner chamber, is actually has a ground matrix chamber of matrix. The matrix contains enzymes for Krebs cycle, proton carriers like NAD plus and FAD plus, mineral sands like ferric, cuprous, and manganese. ADP and ADP and inorganic phosphates also present. Stranded circular DNA also present. 70th ribosome and polysome complex. This is the diagram. Outer chamber, outer membrane, and outer membrane, outer chamber, inner chamber. Here F0, F1 particles on cristae, dicircular DNA in the matrix. Now here we have also many 70th ribosomes, and there is a polysome complex. Uh, Now cristae. Cristae is highly magnified diagram is here. Now the protein comes in the cellular in the inside of cristae through this membrane, and the energy required for this transport of proton is taken uh, by the breakdown of the by Krebs cycle, and it increases the proton gradient inside there. And then this is F1, that is head, and it is extrinsic protein, and it is in enzyme ATP synthetase. F0 is base and is permeable to proton, and it is tunnel protein and also known as proton tunnel. Now, functions of mitochondria is aerobic respiration. Glucose is broken down into pyruvic acid by glycolysis in the cytoplasm. Pyruvic acid enters the mitochondria for further oxidation by Krebs cycle, also known as TC or citric acid cycle. During the pathway, pyruvic acid gets broken down into to release energy and CO2 as a base product. The energy of substrates, the energy released, is used to form excited electrons, that is energy rich electron. The excited electrons are carried by coenzymes NAD plus and FAD plus with the help of protons, and the reduced enzymes that is formed by the NAD plus enter the terminal oxidation inside the cristae. The cristae has a series of electron carriers called electron transport chain. In this, the electrons are arranged in a sequence of increasing affinity for electron but decreasing energy. That is, the electron that will come in the membrane of the cristae will be first enter the electron transport chain. This is the chain. Now if electron comes into the cristae and tries to enter with the cristae membrane, then it is shifted upon according to its energy. Suppose it has a lot of energy, then it would be in A, then it would be in B, then C, and gradually decreasing. Now it is carried out by NADH H plus, that is reduced coenzymes. This lets it enter into the terminal oxidation that happens inside the cristae. and the proton gradients energy is used to for all this. Now the reduced coenzymes head over to the excited electrons to electron transport chain, that was ABC, from A to B then C. And as electrons move from one carrier to another, they will lose energy which is used for active transport of protons. Now as they lose energy while going from A then B then C, they gradually lose their energies, and the energy that is lost is used for the transport of protons inside the crystal, so as to make a proton gradient inside. Now as active transport for protein from matrix to per mitochondrial space, that is the inside of crystal, and this generates a proton gradient, and the proton therefore moves from perimeter to the matrix through F0 F1 particles. Now as see, the energy that was obtained from Krebs cycle was utilized to form the excited electrons. Now excited electrons are carried to the crystal by the help of the terminal reduced coenzymes, and it enters the electron carrier electron transport chain, and as it gets carried on, the energy of that gets loose is used to make a protein gradient inside the that is inside the perimitochondrial space, that is inside of Christie. Now as the protein gradient gets formed, now it would definitely move from high concentration to low, so it will move again from this to the matrix through F0 F1 particles. Okay, now F0 F0 particles, and thus this will activate the enzyme ATP synthetase, which was present in F1 head. And okay, it would just proton would go out with F0 F1 particles, yes, from the here to here in the cell matrix. And the F0 will have ATP synthetase, and ATP synthesis will start again, and from the oxidation of glucose, and that is why it is called oxidative phosphorylation. And that is how ATP gets formed. Now ATP is this one AP, then the triple O, then one, this structure is called AMP, because here only one we have one phosphate structure, so it is called adenosine monophosphate. Then two combine to form adenosine diphosphate, then three combine. This is ATP, that complete. Now next we have nature of mitochondria. Mitochondria is able to synthesize most of the proteins for its use using its DNA. Now some proteins are synthesized by nuclear chains and then given to mitochondria. New mitochondria rises from binary fission, and um, then plastids. Okay, this is some extra information. Now large protoplasmic organelle, then plant, and then animal. Okay. Protoplasm. Hmm. Ah. Plastids. Plastids formed from proplastids. These are undifferentiated plastids. Depending upon functions, proplastids can be of following types: leucoplast, chromoplast, and chloroplast. Plastid is a polymorphic because of three types. Can be simultaneously present in a cell, so it is polymorphic. If we have protoplastids in developing leaves, if less light is given, then it will form Achioplast, that is yellow or colorless chloroplast, since light is necessary for photosynthesis, so it will change its color to yellow or colorless. Now Achioplast is again, if it exposed to light, then it can develop green pigment and become normal like earlier and become green. Now achiolation is yellowing of leaves due to low intensity of light. Now leucoplasts are colorless and helping the storage. These are again of three types: amylo, aleo, and alluro. Amylo, as we know, starch. Now these are again in the form that okay, they will have a protein center and there would be starch layers surrounding it. Now this is potato. In potato, this is eccentric, that is not in a correct manner. Now you see the starch layers form just as the way they are at the beginning, and it keeps going on, and it has a protein center. Similarly rice, but rice is concentric. Now alioplast is lipidoplast, also known as. Now stored soil, it is present in sunflower, mustard, cashew, etc. Aleuroplast is proteoplast, it stores protein. Now if you see a corn, this is one part of the corn that is yellowish color. Now it is a triangle, okay. Now this is just one piece of corn. Now here is, you can see that this is the alluro layer, which is actually yellow in color, and in just the center of it there is a starchy core that is 100% white, and then there's in the side is there is a embryo, and starchy core plus alveolar layer is called endosperm of the cell, okay, storage of the cell. And we had read this in class 10th. Now chromoplasts are the colored and the non-photosynthetic pigments. Okay, pigments, not they are not pigments, but they are actually plastid, of course. Now they are formed from proplastid, okay, and also from for chloroplast. Now, if chloroplast contains both chlorophylls and carotenoids, we know that, and by the breakdown, only carotenoids are left. As we know, lemon is green, okay, but as the breakdown occurs, it becomes yellow. Tomato is in the when it is not ripened very much, it is greenish and reddish, but when breakdown occurs, it becomes very red, and yeah, then it's gradually orange and something like that. Chromoplasts are found in petals to attract pollinators and can be found in vegetables and fruits as well. Now, carotenoids that are present in this chloroplast and which is present in this, okay, chloroplast. Now chloroplast again is another plastid, and it is also a building of helps in the formation of another plastid. And now which chloroplast contains chlorophyll and carotenoids. Carotenoids have two parts, carotenes and xanthophylls. Carotenes are examples are beta carotene now and lycopene, which is present in the red tomato. It is an antioxidant, removes toxin from blood and liver. Xanthophylls are examples are viola xanthin and myoxanthin. Violozanthin is purple and neoxanthin is blue. Now chloroplast, very important. It is green photosynthetic, and it has following pigments: chlorophyll A, chlorophyll B, and carotenoids. It also has carotenoids. Okay, the these are chlorophyll and carotenoids. Chlorophyll again A and B. Now example are containing high chlorophyll are present in the algae, bryophytes, pentophyte. Okay, chlorophyll A and C mixture is found in brown algae. AD is in red, AE is in yellow. Now shape of chloroplasts, it may be cup shaped in chlamydomonas, okay, oval in algae but has different shapes. Now in chlamydomonas it is cup shaped inside it. Eulothrix it is girdle shaped. Spirogyra it is ribbon shaped, and Zyma it is star shaped. Now number of chloroplasts per cell: Chlamydomonas 1, Eulothrix 1, Zygnema 1, Spirogyra 1 to 16, and higher plants is above 16, 16 to 20. Now structure of chloroplast, it is very similar to that of mitochondria, but there are differences as well. It is again a double membrane organelle like that, and outer membrane is smooth and it has porins just like that. Now intermembranous space, that was resembles very much to outer chamber, has filled with fluid and is poor in enzymes, same. Now inner membrane has infolds to form thylakoids just like Christie. but different from that. Now thylakoids are flattened bag-like structures. They are stacked to form grana, and the grana are interconnected with the help of fret membrane or stronal lamellae or integral thylakoids. These are just the names. Now grana are suspended in the stroma. The membrane of thylakoids will have embedded particles, photosynthetic pigments, electron carriers like cytochromes and plastocinin. Cytochromes is common, but plastocinin is different. Okay. Now it will have CF not F1 particles, not F1 or F not F1.